So there's this whole thing called membrane potentials, right?
Uh-huh.
I got it right on the time.
And then I want to understand this down here as well. So, yeah. I kind of think
Didn't want it
it's,
down here.
yeah. Basically just,
Okay.
I
So-
understand that too.
So I'll tell you, this is probably one of the hardest conceptual things
Mhm.
that people hit in this class. So there's this whole thing called membrane potentials. I'm gonna draw a chart and I'm gonna explain it. So bear with me for just 1 second, okay?
Okay.
So positive, negative. And I'm gonna draw a cell. Okay, from the reading that you've done, do you remember where is high in concentration of sodium? Inside the cell or outside the cell?
Outside the cell.
Outside, okay, sodium. Do you know what else is high outside the cell?
Let's see. No, I know that potassium is high inside the cell.
Okay, potassium is high inside the cell, super
And
important.
There's also like chlorine and calcium.
Chlorine, calcium, they're both high outside the cell.
Okay.
So calcium,
Oh, and then magnesium's high inside the cell.
yeah.
Okay.
Typically, for the ones that you're going to be dealing with the most are going to be sodium, calcium chloride, and potassium.
Okay.
Other things are always good to know, but those are going to be the main players in action potentials that you're going to deal with. So, when we're talking about action potentials, we're talking about the charge and the cell. So there's something, you'll see a graph kind of drawn like this a lot, and this is talking about the charge of the cell in reference to the extracellular fluid. Okay? So do you know what charge the cells are typically? If they're positive or negative?
They're negative.
Negative. Why?
Because they're at rest. I don't know why exactly.
No, that's totally fine. Sometimes I'll ask questions that you don't know the answers, but it's good because it lets me know. Because sometimes you know it, and then sometimes it kind of gives me a working place. So inside the cell, there's a lot of negative proteins.
Okay.
Because all the proteins that you see everywhere, they have a negative charge. So that's a big factor, is there's negative proteins. In class, he'll talk about a couple other reasons why they're negatively charged, but I'm going to move on just because I'm going to try to cover all the material that you want to hit.
And potassium moves from high to low, is that why?
So it wants to move from high to low concentration,
Mhm.
but potassium is hydrophilic. Yeah, hydrophilic, so it can't move through the cell membrane by itself, right?
Uh-huh.
So what does it need to travel through?
The protein.
Yeah, so it needs proteins. So we have channels here. Here's a potassium gated channel, okay? Here's a sodium gated channel. And those are going to be the main ones I'm going to talk about right now, because the other ones I'm not going to address for a little bit. I'm going to try to keep it a little simple. So potassium, sodium. Okay. So because the inside of the cell is negative compared to the outside, we say its resting membrane potential is negative. Typically, we kind of average it out, and we're going to say it's negative 70, okay? Because I think that's what it is for neural cells. But yeah, so typically it rests at this charge, okay? But if I did something that opened this sodium gate, where would sodium flow?
Sodium would flow in.
It would flow in because it's high out here. So it's going to flow inside the cell. What would that do to its charge? Make it more negative or more positive?
More positive.
More positive. So we have an event that depolarizes it. Okay? This is called depolarization because right now there's a difference or a polarization between the cell and the outside.
Okay.
So it is depolarized. So that rising phase is called depolarization.
That means it's at work, right?
Not necessarily.
No.
Just depolarization literally just means that it's stopping polarized. Like when you polarize a magnet, that's when you give it its charge. So when it's being depolarized, it's losing that charge and it's going up.
Okay.
Okay? It takes a while. It takes a couple, I think he lectures on this for 2 whole class periods, I think. So it's all right that it's not all coming together yet. But so this is called depolarization. Okay? What is going to happen, though, is after a little while, this sodium gate is going to close. Okay? And I'll explain some of these terms in just a little bit, but so it closes, and as this closes, this potassium gate opens. Okay? So, what's going to happen to sodium?
Sodium will stop going in.
Stops going in. What happens to potassium?
Potassium will go out.
So potassium starts to flow out. How does that affect the charge? Positive is leaving the cell.
It'll go more negative.
More negative. So you get this part, and this is called repolarization, because we're repolarizing it. So depolarization and repolarization. Okay. What happens, though, is that stays open a little bit longer, so it actually even goes further, goes lower than it originally was. And this little part where it goes lower is called hyperpolarization. So does that make sense?
Yeah, that makes sense.
Okay.
Is there like a point where sodium gates shut, or is it different?
What it is right after. It's kind of like I would compare it to a rusted hinge. So it's right after this guy. So this one's called the activation gate. This one's called the inactivation gate for sodium. So sodium's activation gate pops open, and its inactivation gate, I would say, is kind of like a rusty hinge. It takes a moment before it can close. And then when it closes, about that same time, this is also like another rusty hinge. The potassium slowly is opening that whole time. Does that make sense?
Yeah.
Okay. So that's going to be your depolarization, repolarization, then hyperpolarization.
Are some protein channels, do some have 2 gates and some have 1 gate?
Yes.
Yeah.
Sodium's going to be the really important one that you talk about that has 2 gates. So sodium has an activation and an inactivation gate.
Okay.
Then eventually,
And potassium's either just...
potassium just has 1.
Okay.
Potassium only has 1 gate.
What's the point of the 1 gate versus 2 gates?
I don't know, to be honest. My guess, because I'd have to go off of assumption, is it works better that way. I would think it has to deal with how quickly it can react as well as the charges. Okay? So those are some basic things just to get some bearing. I'm going to go back to these gates real quick. Okay. To have these gates open, we have to have some sort of event happen to start it. Sometimes there's things, sometimes there's some gates that are stretch activated, meaning like if you put pressure on it, it opens up the gate, and then it starts a depolarization. So what happens is something starts a depolarization to get everything else going. Okay.
So like mechanical.
So mechanical stimuli can do it. A voltage can do it. There can be
Ligand.
Yeah, ligands. So ligand being just a particle that's floating that binds to a receptor can cause these to happen. There's a lot of different things that can start it. But what happens is, so these gates, it's typically negative inside the cell. So these gates are negative right on the edge, okay? Negatively charged.
Okay, every gate's negatively charged.
Right on the edge right there, okay? What charges the outside of the cell compared to the inside?
The inside's negative, right?
Inside's negative, so the outside would be
Positive.
positive. So we got all these positive charges right here. And that little negative charge is quite nice and happy being close to them, right? So when we have an event on the cell that lets negative charges, it depolarizes the cell, so it becomes a little negative out there.
It's still positive, but a little more negative, right?
Yeah.
Okay, got it.
It's in a very localized area negative. It causes repulsion, which makes that gate fly open.
Okay.
So that's how the gate opens.
Okay, so as it gets more negative, the gate opens.
Mhm.
More positive, the gate closes more.
Yeah.
Okay.
So that's the general idea, because you were asking about how the gates open.
Yeah.
So the general idea is that there's typically a positive charge right there, and then when it switches to a negative, it repulses the gate, opens up, and then you get the sodium flowing in.